thank you to the organizers for having me today, and thank you for the audience for being here. Good evening. Today I'll talk with you about work I've been doing using genomic data to investigate Scots pine expansion in Europe. So first, let me acknowledge the collaborators involved in this project from all over Europe and the different funding agencies, as well as Bohayarvi lab members for feedback, useful feedback on this presentation. So Scots pine, Pinus sylvestris, is a very important model of European forest. As you can see here in green, it has a very wide distribution from Western Europe in Spain to the eastern parts of Asia. It also extends on a very large latitudinal scale. And it's both very ecologically a very important species in Europe and also quite important economically. And so there's a lot of interest in understanding the evolution of Scots pine, its history, how it adapted to different environments. And today I'll focus on on the different population of Scots pine, how their history, how they have evolved in Europe. So as you probably know, there's been in the past climatic oscillations that has affected a lot of forest species. And to illustrate this, you can see on the left side the records of pollen records. The relative abundance of pollen of forestry species pollen sampled in Greece through time. So on the y-axis you have the pollen records sampled, and on the x-axis you have the time. So you go from the recent time on the left to the ancient time on the right. And as you can see, pollen records which reflect the abundance of forestry shift dramatically when you go in the past, and this reflects these events of population expansion and contractions through time. And on the right side, you can see the extent quite far in the south in Europe of the Fennoscandia ice sheets during the last glacial maximum at about 15,000 years ago. And as you can imagine, Pinus sylvestris distribution was quite different than how it is now. For many forestry species, their population was constrained behind mountain ranges in southern Europe, so behind the Pyrenees, behind the Alps or the Carpathians, in so-called glacial refuge areas. So pollen records are very good to collect information about these fluctuations. However, it's not very good to look at patterns at the level of the species because it's not so easy to discriminate pollen from different species. Something that is very good for that, however, is DNA. And you can also use DNA to look for the signature of these population dynamics, expansion and contractions using DNA. So for instance, you would expect that in ancient populations, stable population, you would have a higher level of genetic diversity compared to more recently expanded populations. Well, it's not always that simple, however. Since as has been demonstrated by Petit et al. In 2003, using genetic data collected across 22 forestry species. And here you can see on the map the black circles, which are areas with populations at higher levels of genetic diversity than the average, and in white circles, areas with populations with lower levels of genetic diversity compared to the average. So you can see that on this survey, the hotspots of genetic diversity, the area with higher genetic diversity levels, were north of these glacial refugia, showing that you can can have other processes, in this case, a secondary contact of divergent lineages after the glacial, after the last glacial maximum. And so when you look at the patterns of genetic diversity within a species, you can also look at the genetic structure to try to understand what were the migration routes since the last glacial. And for Scots pine, for instance, here there is a survey of genetic data using mitochondrial markers that shows the genetic diversity and structure of Scots pine in Europe. And you can see that the structure is very weak. Most of the samples in central, to some extent, northern and southern Europe as well, share very close alleles, actually the same haplotypes. And you can note that there are two divergent groups of populations in Italy, in northern Italy, in a different shade of green, and in Spain in red here. So using this data, Chedly et al. Have been able to infer post-glacial colonization routes. However, one limit of their data and analysis is that it relied on a small number of markers, and so they might have missed some finer details of the story of Scots pine. More recent work has been conducted using genome-wide genetic markers, CIPS data. So here is an illustration of what has been done by Chedly et al. In 2020, looking at genetic diversity across more than 10,000 SNPs in Europe. And you can see that the genetic diversity, like there is very little genetic structure across the sampled populations. However, you can see also that in the south, the Spanish population seems a bit divergent from the remaining populations. There has been also other studies, such as Allele et al. In 2021, but both these studies, so they identify the same patterns of very low genetic structure across populations of Scots pine in Europe, but they haven't actually surveyed the populations in the glacial refugia, except for the Spanish population. But so we still lack a bit a good picture of these populations. And so basically, this is where we try to fit in. We try here, we're looking at the genome-wide genetic diversity of Scots pine, specifically looking also into these glacial refuge areas in Italy, Spain, and the regions. We're trying to look at where are the hotspots of genetic diversity, and using patterns of genetic variation across Europe, we try to also identify the colonization routes. So basically, what are the origin of the different European populations of Scots pine, and especially we try to put them in relation to these glacial refuges. Okay, so a brief overview of the workflow used here. So we are using 20 populations, 12 of which, are actually coming from Chedly et al. 2020, and we are adding 8 new populations. So one distinction between these two sets is that the 12 populations from Chedly et al. Was based on markers identified using megagametophile tissue, meaning haploid tissue, dissected from the Scots pine seeds, whereas we are here adding sets that was DNA that was extracted from needles. We then use an exome capture strategy using the same bait set, so we can really combine these two data sets. And then we followed the standard bioinformatic pipeline for this kind of data, and we ended up with 25,000 SNPs for downstream analysis. So first, if we take a look at the genetic structure of Scots pine, of these populations that we have collected of Scots pine. So we can do that using a principal component analysis. And first, I want to here show you where are these populations sampled in glacial refugia. So you have Italy on one side, Spain on the other, and finally Carpathian. So they are quite well diverged from each other. You can see that Italy is basically driving most of the variation on PC1, whereas the Carpathian samples and Spain as well as all the other samples, are actually distributed along the principal component too. Actually, if you look at the nature of these other populations, you can see that there is a clear geographical pattern going from southwest Europe to northeast Europe. Interestingly, you can see that most samples from Eastern Northern Europe cluster closely with the Carpathian populations, and actually even Northern France actually is closer genetically to the Carpathian populations than to the Southern France ones. So in a second analysis, we use the results of these PCAs to make inference about the ancestral proportion of the individuals in our samples. And we use the tool called eigmix. And so basically this really relies on the PCA, and we have set the three main groups that we are interested in. Spain, that we have put with southern France, Italy, and the Carpathian populations in three sets. And we use the 32 first species of the PCA. What we obtain from this is this plot, which shows on the y-axis the proportion of shared ancestry with each one of these three groups identified, and then on the y-axis samples. Basically, you see the same patterns where you have Spain on one side and the southernmost population we sampled in France, so that they shared their ancestry quite well. And then Italy on its own, and finally the Carpathian. So the northern populations from Europe and eastern populations from Europe, as well as the northern France populations sharing all their, most of their ancestry with the Carpathian populations. So we also looked at the allele frequency distributions in these different groups of samples. So here what you see is a plot which shows on the x-axis the minor allele frequency, and on the y-axis the amount of sites that we have collected, and it's a log scale. So the first thing, and then on each line that you see is one of the groups that we have, group of populations that we have collected. And the first thing that you know that I can say about this plot is that in all groups there is actually an excess of rare alleles. And the second thing is that there is in the Italian population an excess compared to the other populations of common alleles, which here so you can see here on the right side of this plot, the common alleles. And then it's followed by the Spanish population, but that is actually very close to the other populations.
2 minutes left.
Okay, so what we did also was to look at patterns of allele sharing, of rare allele sharings. So basically here we've defined our three groups in a reference panel and looked at how each individual had shared allele with these group of populations. And we focused on rare alleles, meaning that contains a higher proportion of recent mutations. So basically this gives us an insight into the more recent population structure. And it's also interesting since we have so many of these rare alleles. And so if I go directly to the results that you obtain, it's actually very consistent with the other analysis where we see that most populations from Northern Europe, Eastern Europe, so each curve here is the pattern from a single population, while they share most of their allele with the Carpathian populations here in blue. Then comes the southern France and Spanish population, and finally the Italian population. And so this is the common trend in all these analyses and also here in these plots. And so finally, we did also demographic, an analysis of the demography of the species. And so very quickly on the y-axis, you have the effective population size, on the x-axis, the time from present to past. And what you see are the changes in population effective size through time. This analysis done at the level of the species shows that there has been continuous expansion of the species in Europe, but these are ancient ancient events of demography. If you look at the level of the population, you really see the same trends, ancient expansion, perhaps except Italy, which is the most stable population that we have in our data. So basically the conclusions are that while it remains challenging to use genome-wide data on conifer, it's been improving quite fast. And on the biological aspects, so the genetic diversity of Scots pine is very homogeneous, two divergent areas as identified previously in Italy and Spain. And then the remaining populations are actually related a lot to the populations from the Carpathian areas. And this is in quite a contrast with other species of conifers, which have more structure basically. And thank you again for your attention, and thank you to the, again, collaborators of this project.